Dear students, today we are going to discuss about the structure, morphology and the reproduction of ginkgo alien members. This group, Ginkgo allis, came into existence from the Permian, and this group is divided into two families, namely Ginkgoaceae and Trichopitiaceae. There are 16 genera and many species distributed all over the world in the Jurassic period. From the Cretaceous onwards, this group slowly fading away, leaving a single living representative, that is Ginkgo biloba. The characteristic feature of this order, Ginkgo allis, are branching trees with clearly marked long and short shoot, and the leaves which are simple and variously lobed with the dichotomous venation. Wood is pycnoxylic, and the male sex organs that are found in the axil of the leaves in the short shoot, and this male reproductive organs are catkin-like, bearing many microsporangiophore. Each microsporangiophore having 2 to 12 microsporangia. Sperm has got a spiral band of flagella, whereas the female reproductive structure also axillary, and it is having 2 to 10 ovules in the terminal part of the branch. The ovule having a outer fleshy layer, middle stony layer, and then again the innermost one is the fleshy layer. Now you are going to see the morphology of Ginkgo biloba. Ginkgo is one of the oldest living seed plant. That's why it is truly known as living fossil. This plant, Ginkgo biloba, came into existence from the Permian onwards, and this plant is also known as maidenhair tree because the leaves of this plant is very much similar to those of adiantum, and these leaves are deciduous. The plant, it is almost pyramidal in shape, about 30 meter in height. The shoot apex of Ginkgo biloba is one of the primitive type found elsewhere only in the cycas, and this shoot apex has got a shallow cup of apical initials, and that give rise to a core of cells, and there is a flanking region. The core consists of 3 regions: the subapical initials, which are having small cells which are densely strained, whereas the next zone, it is a central mother cell zone where the parenchyma cells are large with many vacuoles, and they are take up the light strain. The third region is the pith rib meristem, which is later on give rise to the pith. The cells of the flanking region is partly derived from the apical initial and partly from the central mother cell zone. Ginkgo biloba, it has got a two shoot system. One is the long shoot or shoot of unlimited growth. The second one is spur shoot, or otherwise known as dwarf shoot, and which has got a limited growth. The long shoot, it is grow very fast, up to 30 centimeter in a year, whereas the short shoot of 2 to 3 centimeter, it take up several years to develop such a small shoot, whereas the short shoot may turn to be a long shoot, or rarely the long shoot terminated into a short shoot. The difference between the long shoot and the dwarf shoot lies only in the rate of cell division in rib meristem, and this difference in rate of cell division in turn is activated by the concentration of axin. If there is a high concentration of axin, the division is very fast and it producing a long shoot, and if there is a low concentration of axin, it producing a short shoot. The leaves which are found in the short shoot are entire, whereas leaves which are found in the long shoot are typically bilobed, hence the specific name ginkgo biloba. And the leaves are simple and it has got a long petiole, and the venation is open dichotomous, and the petiole has got two endark vascular trays, and each one entering into the one respective half of the leaf. The internal anatomy of the leaf shows upper and lower epidermis, and it is thickly coated with cuticle. The mesophyll is differentiated into palisade and spongy parenchyma. There are many mucilage cavities found in between the veins. The vascular bundle has got a sclerenchymatous bundle sheath. The protoxylem is endark. The stomata confined to the lower epidermis, and they are haplochilic. There are 4 to 6 subsidiary cells around the God cell, and in this subsidiary cells there is a papillae-like projection which is arching over the God cells, and these stomata are slightly sunken. The stem of ginkgo biloba having distinct epidermis, parenchymatous cortex, vascular cylinder, and parenchymatous pith. The epidermis is later on replaced by the periderm. The cortex having many tannin cells. The vascular cylinder having endark protoxylem. The secondary wood is pycnoxylic, and it consists of only tracheids and the rays which are seldom one cell wide. The pitting in the radial wall of the tracheid are biseriate, opposite, circular, and bordered. There are many crassulae are seen in between the pits. Many mucilage cavities found all over the plant, not only in the vegetative parts, it is also in the reproductive structures. My dear students, now we are going to discuss the reproductive structures of ginkgo biloba. The plant is dioecious. It is difficult to distinguish the male plant from the female plant. Male plant has got XY sex chromosome, whereas the female plant has got XX sex chromosomes. The male plant having male reproductive structure in the axil of the leaves of the spur shoot. Each male reproductive organ having a main axis with many lateral microsporangiophore. Each microsporangiophore has got a terminal knob containing a mucilage cavity, and there are two pendulous microsporangia. This microsporangia are many wall layered and it degus by means of longitudinal slit, whereas in the female plant the ovules are borne on a stalk that is found in the axil of the spur shoot. There is a stalk and it is bifurcated its terminal part, and each lateral axis has got a single ovule, and below this each ovule there is a shallow cup like structure that is known as collar. Some may consider this collar probably an reduced sporophyll, because each stalk of this ovule receives vascular bundle, it is double the number of ovules, and so we consider the collar is an reduced sporophyll. Similarly, the mucilage cavity that is found in the microsporangiophore is nothing but an abortive microsporangium. That means the ginkgo biloba plant is derived from a plant having microsporangiophore having more than 2 microsporangia like Baira. Each ovule consists of a single integument, and that integument is differentiated into 3 layers. Outer fleshy layer consists of very large parenchyma cells with many mucilage cavities, whereas the middle layer is the stony layer, consists of isodiametric cells, whereas the innermost layer, that is again a fleshy layer, consists of many parenchyma cells, and after fertilization the innermost layer, it forms a paper like structure. And in the beginning of development of female gametophyte, there is a single functional megaspore mother cell that undergoing meiotic division to produce 4 megaspores, and among these 4 megaspores only the lowermost one is the functional megaspore, and that divides to form the female gametophyte. In the time of pollination, the pollen grains are drawn into the ovule through wind currents. At the same time, there is a pollination drop secreted from the ovule, and it is come out of the ovule and stay there in the micropyle. Once the pollen grains caught into this pollination drop, if the pollination drops dries up, it is drawn into the pollen chamber. Simultaneously, there is a development in the female gametophyte. The functional megaspore, the nucleus of this undergoing repeated mitotic division. Roughly there are more than 8,000 nuclei are formed without any wall formation. This stage of development of female gametophyte is known as free nucleus stage of the development of female gametophyte, and afterwards the wall formations start from the periphery and the entire female gametophyte is cellular. There are 2 to 3 archegonia per ovule. Each archegonium consists of a spherical egg cell, and there is a ventral canal cell and a neck of 4 cells. Once the pollen grains are released from the microsporangia and it is enters into the ovule, the remaining development of male gametophyte, it is taking place inside the ovule. That is, now the pollen grain consists of 2 prothallel cell, 1 generative cell and 1 tube cell. Now the wall of the pollen grain that is found in the side of tube cell is forming a long tube and highly branched at the terminal part, and it is completely covered over the nucellus. Now it acts as a hostorium and it absorbs the food material from the female gametophyte. Simultaneously, the generative cell divides into 2 cells. One is the spermatogenous cell, the other one is stalk cell. The stalk cell is like an stalk of an antheridium. The spermatogenous cell divides into 2 sperms, and each sperm is a top shaped structure with 3 bands of flagella, and it is a motile sperm. Apart from the cycas, only in ginkgo you will find the motile sperms. Then after this development, the sperms are ready to enter into the archegonium. Now the pollen produce a pollen tube towards the prothallel side, and the sperms enters into the pollen tube. Once the pollen wall get ruptured, the sperms are released into the archegonial chamber. Now once the sperm touches the neck of the archegonium, it loses its flagellated sheath and only the nucleus alone enters into the archegonium. Among the 2 sperms, one enters into the archegonium and the nucleus of this fused with the egg nucleus and it forms a diploid zygote. The embryogeny of Ginkgo biloba starts with the zygote nucleus divided into many nuclei without any wall formation. There are more than 256 nuclei are formed. Then the wall formation start from the periphery, and it is now the entire zygote is cellular. After this, the cells which are found in the micropylar region is elongates and it forms a massive suspensor, and the cells which are found in the lower part, that is chalazal end, that forms a embryo that is primarily with 2 cotyledons and the shoot apex. The root apex, it is found in the micropylar end. So the mature embryo has got a suspensor with 2 cotyledons. This mature seed, it may be shed before or after fertilization. When it is sheds from the parent plant, there is an outer fleshy layer in the seed, middle stony layer, and the innermost fleshy layer, now it turned to be a watery structure. Many considered this Ginkgo ellis is one of the primitive gymnosperms like psychedelian members, so in which it shares many primitive features like the motile sperms. But Florin concluded that it is The Ginkgoalis is like Coniferales, Auddeyt, Taxalis. It is one of the natural group found in the Coniferopsida, and all these four groups are evolved from the Permian in a parallel way.